Hello, my name is Steve White. I'm professor of pharmacology and toxicology at the University of Utah in Salt Lake City. I also direct the anticonvulsant drug development program, which since 1974 has utilized a variety of animal seizure and epilepsy models for the early identification of anticonvulsant activity. The current era of antiepileptic drug discovery was ushered in 1938 by Merritt and Putnam when they first identified phenytoin from a series of over 700 hydantoin analogs using the cat maximal electroshock seizure model, which is a model of generalized tonic-clonic seizures. In addition to the maximal electroshock seizure, the anticonvulsant drug development program, as well as other anti-epileptic drug discovery programs worldwide, incorporate a variety of different animal models of generalized and partial seizures. The ultimate goal of this initial identification is to provide the sponsor with sufficient proof of concept data that they can file for an investigational drug application and enter their compound into clinical trials. This slide shows that since 1912, using this rational approach, a number of new anti-epileptic drugs have come onto the market. More recently, since 1993, you can see a variety of different drugs called the third generation anti-epileptic drugs have emerged using the same approach or a slightly modified approach to that which has been incorporated over the last 3.5 decades. The most recent anti-epileptic drug to be approved is retigabine or ezogabine, depending on which side of the Atlantic you sit on. And an important point that I'd like to make, although you can't really see it on this slide, is that the current approach does identify rather mechanistically distinct anti-epileptic drugs. In addition, there are a number of different drugs that are currently in the pipeline and will hopefully emerge over the course of the next 1 to 5 years. And again, like the third generation drugs that have come out since 1993, the drugs that are currently in the pipeline that have been identified using this approach are very mechanistically different from each other, including 2-deoxyglucose, which affects intermediary metabolism, ganaxolone, a neurosteroid, huperzine, an NMDA antagonist, NAX 5055, or a similar analog as a galanin-based compound, and so on. The ultimate hope is that these drugs emerge successfully from the pipeline as opposed to crashing throughout their developmental process and make it to the patient for the treatment of epilepsy. What the current approach doesn't really address is the adult with highly refractory partial onset epilepsy. As you well know, approximately 30% of patients with epilepsy still remain unsuccessfully treated with the currently available therapies. Despite the success of this systematic approach, there still remains a substantial need for several different patient populations, including the child with catastrophic epilepsy, the elderly, the patient with highly refractory epilepsy, which despite multiple drugs still continues to suffer from seizures. Moreover, the current approach is not addressing the patient's comorbidities that they might experience. In the future, we'll hopefully look towards identifying drugs that can not only be effective in treating the seizures, but also address some of the comorbidities. So in summary, the current approach has been found to identify clinically effective anti-epileptic drugs. Unfortunately, despite the success that we've enjoyed, there still remains a substantial need for the patient with highly refractory epilepsy, the child with catastrophic epilepsy. And we need to begin to think about finding therapies that may be useful for the treatment of comorbidities associated with epilepsy. As I'm sure you're all familiar with, there's also current effort to try and identify the patient that may be susceptible for developing epilepsy and to initiate therapies that could either prevent the development, such as an anti-epileptogenic therapy, or modify the seizure severity and the seizure type once epilepsy has been diagnosed. I believe that there is a way forward. However, I think it's going to require an investment both in time and resources. It's going to take communication between the basic and the clinical sciences, and all working together to move forward to the next generation anti-epileptic therapy or any therapy for the treatment of epilepsy. Thank you very much.